How do you know if a specific protein is present in a sample? The main way that you'll know in biochemistry is with the use of antibodies, which are little proteins that we'll talk about that recognize specific features of other proteins or other molecules. Much more details later, but basically they recognize very specific parts of other molecules, and therefore you're able to detect specifically the presence of those other molecules. So not just are proteins present, but is this specific protein present? Not just is Waldo there, but is Waldo's hat there? This is going to allow you to do things like see if a protein is present in the sample. One of the main ways that we'll do this is with the Western blot. So you run the proteins on an SDS page gel, transfer the proteins to a membrane, and then probe the membrane with antibodies to see if the protein is present and how much. You can do something similar in a plate format, which is something called an ELISA or enzyme-linked immunosorbent assay. And if you want to get fancy with your Western blotting, what you can actually do is you can see if different proteins are interacting with one another by pulling down one protein. So using an antibody to kind of take out one protein and see what it's bound to by then probing with the Western blot for whatever you're looking for that it might be bound to. So we'll get more into the details, but these are the kind of the main strategies that we'll talk about using with antibodies, our Western blot, our Co-IP, and our ELISA. Let's dive in. Let's start by thinking about what is an antibody. An antibody is just this little protein that's made by the immune system, and it recognizes different features of other molecules. We'll talk in the terms of proteins for now. So let's just say that an antibody is a little protein that recognizes another protein, but it actually recognizes a specific part of another protein. And note too, I'm going to talk about protein just in terms of simplifying things, but you can also have antibodies that will recognize things like specific RNA or DNA features. And you can have antibodies that recognize things like phosphorylated forms of proteins or non-phosphorylated forms of proteins. The reason why you can have such a variety of things is because the antibody is actually only going to be binding to a very specific portion of the molecule which it recognizes. Note that when we say recognize, all that really means is that it binds specifically. So when it binds, it's going to be binding through non-covalent interactions. These interactions are going to involve our good old intermolecular forces. They're going to involve things like your dipole-dipole interactions, and importantly, things like your hydrophobic interactions. So this is just showing you a binding site for the antibody binding to the antigen. So the antigen is the thing to which the antibody binds. And more specifically, the epitope is the part of the antigen to which the antibody binds. So more on that in a second. But here is a binding site. And you can see that it's colored based on hydrophobicity. And this yellow is going to represent hydrophobic parts. So remember, our hydrophobic parts are going to be those parts that are nonpolar, those parts that water excludes. And so typically you're going to see them clustered in the center of a protein. If we look at the structure of this antibody, and we see this is kind of like the antigen binding site, so this is where it's going to bind the antibody, we can see that it's kind of hiding some hydrophobic residues in the center. You can imagine that if you had water, the water would be all tied up in here in this little channel, and that would decrease its entropy. Binding to the antigen, however, well, now this is going to allow you to free up the water that was tied up in there, and it's going to therefore increase the entropy of the water, driving the interaction. So this is just showing you the conformation of the antibody after it binds to the antigen, showing you that when the antigen binds, you actually expose more of this hydrophobic surface, but it is then being complemented by the antigen, so you get this induced fit that's releasing a bunch of trapped water, increasing your entropy, and this is going to contribute to very high affinities. So antibody-antigen interactions are going to be on the order of like 10 to the minus 8 to 10 to the minus 12 KD in terms of their binding affinities. And so remember, KD, the smaller the better, because that tells you the concentration at which the concentration of one partner at which half the other is bound. And so if we're talking about like 10 to the minus 9th, that's going to be your nanomolar. 10 to the minus 12, that's going to be your picomolar. These are very, very high affinity interactions, much higher than the affinity of, say, your enzyme substrate interactions. but not quite as high as the biotin-avidin interactions that we discussed. What we were looking at there was just kind of looking at this one little portion of the antibody. And so we can think about the antibody as being kind of splittable into multiple portions. And so we have this portion up here. This is like your FAB, your Fab domain. And then this is your FC, the fragment crystallizable. Sometimes you might see this kind of top portion, these arms. So if you think of a Y, you might see the arms of the Y called FAB, fragment antigen binding. And then the FC would be the fragment crystallizable, which is kind of the base of the Y. And then the arms of the Y would be your Fabs. Antibodies are going to consist of both variable and constant regions. So the variable regions, these are the unique regions that are going to allow antibodies to recognize specific things, and they allow different antibodies to recognize different specific things. And then you have your constant regions, which are going to serve as adapter points so that multiple proteins can work with these antibodies and bind to these antibodies and do things like carry them throughout the blood. What we're looking at here is what we call an IgG antibody. There's also IgM antibodies, where here, that kind of generic adapter part is used to link them together into these pentamers. So you have 5 copies. And this is going to then allow you to get avidity, which is basically where you have one antibody bind and it kind of helps the others bind because it increases the local concentration. And so different antibodies are made at different times in your body, and they're made at different places, and they have different functions, and we're not going to go into the immunology, but these antibodies are going to recognize specific features. The specific features are going to be what we call epitopes, and those epitopes are on antigens. So if you want to think of a game of Where's Waldo, you can have multiple antibodies that all recognize Waldo. In that case, they would all have the same antigen. Waldo would be the antigen. However, they could recognize different features of Waldo. So maybe you have an antibody that recognizes its hat, an antibody that recognizes its elbow, and an antibody that recognizes its toe. These would have the same antigen, but they would have different epitopes that they recognize. So the epitopes would be like the puff on the hat, the crease of the elbow, and the tip of the toe. Note too that if you had a mix of these antibodies that all were recognizing Waldo, but different parts of Waldo, this is an example of a polyclonal antibody. Whereas if you had only one of these antibodies, so you'd have like a lot of copies of the one that recognizes this hat, or a lot of copies of the one that recognizes this elbow, or a lot of copies of the one that recognizes this toe, that would be an example of a monoclonal antibody. Antibodies are made up of 4 chains, 2 heavy chains indicated as H, and 2 light chains indicated as L. So you'll have 2 copies of the H chain and 2 copies of the L chain. So this is going to be an example of a heterotetramer. You have 4 chains, and they're not all going to be the same. The antigen binding site is going to be kind of at the tip, and it's going to be made up of contributions from both the heavy and the light chains, from portions of those heavy and light chains that vary. And so we call these variable regions. So we have the variable heavy portion of the heavy chain, we have the variable portion of the light chain, and then we have the constant regions of both. And so the antigen binding site, the part that recognizes the epitope on your antigen, is going to be made up of contributions from both the heavy and the light chain. And therefore, what it recognizes is going to depend on the sequences of those heavy and light portions of that specific region. The constant region is going to serve as the adapter that's going to allow different molecules to kind of grab onto the antibody. Keep these constant regions in mind because this constant region is going to be constant for the animal that makes it. So it'll be constant, say, for a rabbit, if the rabbit made the antibodies, or it'd be constant for a mouse, if the mouse made the antibodies, or it'd be constant for a human, if the human made the antibodies. This is going to allow us a way to use other antibodies, which we call secondary antibodies, to recognize the presence of an antibody. So recognize the presence of an antibody from a mouse or from a human or from a cow or a rabbit just by recognizing the constant region. So this is going to come into play when we talk about how we can actually put these antibodies to use and use secondary antibodies to help us recognize the primary antibodies. But going back to our structure, we see that you have these 4 chains, and you can see that we have these S's. These are indicating disulfide crosslinks. So remember when we have a disulfide crosslink, those are going to be consisting of 2 cysteine residues kind of linking up. This is an oxidized form of cysteine. And therefore, if we were in the lab and we were to add our reducing agent, so if we were to add something like DTT or beta-mercaptoethanol, that would break up the structure of the antibody. And in our bodies, well, we have reducing agents inside of our cells. And so typically inside of our cells, these antibodies want to be very sturdy. But in the oxidizing environment outside of cells where these antibodies are secreted into, having these disulfide bonds, these covalent linkages, is going to help hold the antibodies together. So this is the structure of an antibody. We've got those 4 chains. 2 of the chains are going to be the heavy chain, and 2 of the chains are going to be the light chain. We have the variable regions of the heavy and of the light chains are going to be kind of at the tips of our Y. And then we have the constant regions, which are going to be kind of the base of the Y. And those variable regions are going to be variable because of having different amino acid sequences. And if we think about how we can get different amino acid sequences, well, we have to edit the DNA. And it turns out that this is what immune cells do. Antibodies are going to be made by the immune system. They're made through a kind of trial and error process. What happens is that they're made by cells called B cells. These B cells all start with having the same genetic repertoire, but then they kind of splice together their DNA in different ways. When they splice together their DNA in different ways, they basically commit themselves to making one specific antibody for the rest of their lives. And so any antibodies that are made by that B cell are therefore going to be of that specific type. This is just a random process. So they might make a good choice and they might make a bad choice. And well, you can't really tell if you made a good choice until later on. You can tell from the beginning whether or not you recognize yourself. And if you do recognize yourself, you're going to kill it so that you don't kill yourself by recognizing and therefore attacking yourself. But you won't know if you're good for something else. Are you able to recognize something foreign, something specifically foreign, unless you're actually confronted with that foreign thing? And so you have these clones kind of just wait around, and they're just kind of testing the waters. And if something foreign comes in, and an antibody happens to bind it because it randomly happened to make a choice that happened to make it have that antibody that recognizes that thing, it'll get selected for, and then you'll make a lot more copies of it. And they're making a lot of copies of it. You can think of it as like a clone. And so you would have monoclonal if you only had antibodies coming from that one B cell that was making that one type of antibody. And then that B cell would then kind of be able to produce antibodies for the body and make more of that cell and all that great stuff. And so sometimes in the lab, you'll have to choose between using a monoclonal antibody or using a polyclonal antibody. Not going to go into the details here, but the monoclonals are going to typically be more specific as long as that is you have one good antibody, but the polyclonal are often going to be cheaper, and they're going to often allow you to have a higher sensitivity because you have more kind of places on Waldo that you can bind. So instead of only being able to bind like one place on Waldo per Waldo, you can bind multiple places on Waldo per Waldo. This is going to allow you to detect lower levels of Waldo with a higher amount of signal. So again, these are just technical details that I'm not going to go into in this video, but I'm happy to tell you more and direct you to resources that will tell you more. But for now, let's just focus on kind of how we can use these antibodies. One of the reasons why I brought up the whole monoclonal versus polyclonal thing is because antibodies can be really expensive to make. And it usually involves using various lab animals. There are, however, moving towards models where people can make antibodies without the use of animals, and so that's probably going to be the way of the future. But for now, animal production is still a large kind of component of the process of making antibodies, allowing animals to do that kind of trial and error process in order to find the antibodies that work and make more of them. Because this is such a long, laborious, expensive process, scientists don't want to go through that process on their own, and typically they're going to outsource it to various companies, and those companies are going to charge them a lot of money. And it's just not technically feasible in order to make an antibody for every single thing that every single scientist wants to study. So instead, what scientists often do is they introduce an epitope onto the thing that they want to study. So remember, an epitope is the part of the antigen to which the antibody binds. If you add an epitope that you know an antibody will recognize to a protein, so you add a sequence to a protein, like as if you were to add the hat, Waldo's hat, to your protein of interest, because you know you had an antibody that recognized Waldo's hat, well, now you're able to recognize your protein of interest. And a different researcher might put that hat on their protein, and that protein would be different. And now the antibody would be able to recognize indirectly the presence of both of those proteins. So different researchers can use the same antibody to recognize different things, as long as they put the epitope that the antibody recognizes on the thing that they're interested in. So we can stick this epitope onto various proteins in order to detect the presence of those various proteins by detecting the presence of the epitope that they're attached to. Instead of adding Waldo hats, often what we're going to be doing is we're going to be adding things like flag or HA or MIC. And then we can use things like anti-flag antibodies in order to detect the presence of the molecule to which we have attached that tag. And so remember that really all we can attach in terms of tags protein-wise is this just going to be a little sequence. So a flag tag is just, say, going to be a short sequence of amino acids that then we have an antibody that can directly recognize. And if we have an antibody that can directly recognize it, that's going to then allow us to indirectly recognize the presence of our protein. So now let's discuss how we can actually recognize the presence of something. Let's start by thinking about where we might be wanting to recognize the presence of a protein. In the case of having proteins in cells, say, you might be able to use an antibody that is going to just bind to cells that you fixed. So you kind of immobilize the cells and you make them permeable. So you add a chemical that's going to allow things to go into the cells. And then you can add an antibody that's going to go into the cells. And as long as you have a way to detect the antibody, you have a way to detect things within the cells. So this is called like immunocytochemistry or immunostaining, where you're kind of staining for the presence of various proteins inside of cells. I'm not going to talk too much about that. I'm going to be talking more about in what we do if we have a sample that we take out of cells and then test it. And so that would be like a Western blot, but the same sort of principle apply, including the fact that you often are going to involve secondary antibodies. So I want to say a few words about secondary antibodies, and then we'll go a little more in detail into the methodology of our Western blot, our ELISA, and our Co-IP. So secondary antibodies. So I kind of hinted at this before when I was talking about we had these constant regions, and these constant regions were going to be constant for the animal that produced it. So if you had antibodies made by a mouse, they would have a constant region that was mouse specific. And if they had antibodies made by a rabbit, you'd have a constant region that were rabbit specific. This is going to be helpful because this is going to allow you to have antibodies that will recognize an antibody made by a mouse or an antibody made by a rabbit or an antibody made by a goat or by a human. You can have antibodies that recognize the species that the antibody was made from. So the primary antibody is going to be the antibody that directly recognizes the thing that you're looking for. Ideally, this is going to be recognizing some specific feature of your protein, but if you can also have one that recognizes like a tag that you put on your protein. This is then going to allow you to have something that binds to your protein. It binds to that variable region, and it still has this constant region. Your secondary antibody is going to have variable regions that recognize the constant region of your primary antibody. And these secondary antibodies are going to be conjugated to something that's going to allow you to see the presence of the primary antibody, which is showing you the presence of the thing that you were looking for. So for example, if we call this protein poly, we would have an anti-poly antibody. And maybe that anti-poly antibody was made in mice. So this would be a mouse anti-poly antibody. In order to detect the presence of the poly, we would use this primary antibody, the anti-poly antibody, to get this antibody to bind to poly. But in order to actually detect the binding of the antibody to the antigen, which is telling us that the antigen is present, so it's telling us poly is present, we need it to actually be able to talk to us. And so to get it to talk to us, we're going to use a secondary antibody. The secondary antibody, so say maybe you had a goat antibody, that was anti-mouse. And so you would have a goat anti-mouse antibody that will recognize the constant region of the mouse antibody. And this is then going to allow you to detect the presence of the primary antibody because your secondary antibody is going to be conjugated to something that you can see. So an indirect way in which you can amplify the signal, because now you can have multiple of these secondary antibodies binding to the primary antibody. Even more, if you have an enzymatically linked reaction, such as your horseradish peroxidase or your alkaline phosphatase, these enzymatic reactions, because they're catalytic, you're going to get an increase in your signal. You're going to get a high amount of signal. You can also imagine that because you can have lots of different variable regions connected to that same constant region, but you can have the same secondary antibody recognize all of those different antibodies because they have the same constant region. You're therefore able to use the same secondary antibodies for a lot of different primary antibodies, and this is going to dramatically decrease the cost of them. So your secondary antibodies are often going to be pretty cheap, whereas your primary antibodies are going to be more expensive. And this also gets back to why you don't want to just label your primary antibody, because if you label the primary antibody, that's going to make it even more expensive. And then it's only going to be useful for that one specific purpose. Whereas by conjugating to these secondary antibodies, you can make these secondary antibodies more broadly useful. So this use of secondary antibodies is going to be very, very common, except you might see like primary antibodies be conjugated if it's a primary antibody for a protein that lots and lots of people want to look for, or maybe an epitope that lots and lots of people want to look for. So you might see like already conjugated anti-flag antibodies or something like this that are already attached to a fluorophore or to an enzyme. I don't need to worry about that, but just know it's something you might come across in the lab. For now, let's just think about kind of a more typical situation where you're using a primary and a secondary antibody to detect the presence of a protein. And again, I'm going to be talking in terms of a protein as our antigen being a protein, but your antigen can also be either like a specific modification of a protein. It can even be a molecule that's not a protein. It can be something like a sugar. It can be something like a specific shape of something, lots of different options, but we're just going to think in terms of a protein to make our life simpler. Speaking of proteins, how do we analyze proteins? Well, one of the main ways that we analyze proteins is through an SDS page. So remember in an SDS page gel, we unfold, so we denature proteins. We do this using heat and a detergent called sodium dodecyl sulfate, or SDS. This SDS has this long structure has this hydrophobic tail that's going to glob onto the hydrophobic parts of the protein that are exposed when you heat up the protein. And it's going to have these hydrophilic heads, which is going to keep the protein soluble. These heads are going to be negatively charged. This is going to help keep the protein linear, and it's going to help promote the travel of the protein to a positive charge. So you can use electricity to create a charge gradient that is going to drive your proteins through a gel made of polyacrylamide, so it's like mesh. The bigger proteins, the longer proteins are going to get caught up more on their way down the gel, and so they're going to travel slower. And so if you were to turn off the gel, you would see that they were higher up. So we're going to run our SDS page gel, and we'll run it of different samples. So maybe you had samples from different cell types, or you had samples from the same cells over time. These samples will contain different amounts of different proteins, and you want to detect the presence of various proteins and the abundance of those proteins. So you take your samples, you run an SDS page gel, and now you want to use your antibodies. If you were to go and try to probe these gels, well, these gels are very fragile, and so your gel might tear and not be very amenable to probing. You also might have the problem where your proteins are actually going to diffuse out of the gel. And so remember, you just have this kind of like gel mesh. And there is right now, there's not like a driving force of the electricity to get the proteins to want to move. And so the proteins are just going to kind of freely diffuse a little. And so they'll just come out of the gel if you were to let them. And so that's why we need to transfer them to something sturdier, something that we can use more long term. And so what we're going to do is we're going to take those proteins that are trapped in the gel and we're going to take them out of the gel and onto a membrane. Once again, we're going to use charge to help us do this. But in this case, we're going to be using charge in kind of like vertically. So we're therefore taking the proteins out of the gel and onto a membrane. Now we want to go ahead and probe the membrane. We want to take those antibodies and see what's present. But if you were to go and add antibodies right now, you would see a ton of noise. This is because the reason why your proteins are kind of sticking to that membrane is you can think of the membrane as a bit like duct tape for proteins. So it'll bind kind of non-specifically to proteins. And well, if we think about antibodies, antibodies are proteins. So if you just add antibodies to a membrane, they're going to bind. So instead, we've kind of got to block it. And so if you think about duct tape and you think about, okay, well, you have parts, you have your molecules that you want bound to the duct tape. Now, if you were to add some sort of lint or something, just to kind of hide the rest of the sticky parts, that's going to make it so that only if you add something that binds to what's already bound, will you be able to see a signal. It's not going to be binding non-specifically to your membrane. Often what we're going to be doing is we're going to be blocking it with a generic protein. So you might see blocking with, say, like casein, or with milk is a common one. So it's like skim milk, or sometimes you might see BSA, various proteins used to kind of hide the rest of the membrane so that all the binding that you'll see is going to be specific to the proteins that you want to detect, the proteins that were actually present in your sample, not the proteins that were just present in the solution that you poured on afterwards. Now that you've blocked the membrane, you're going to add your primary antibody. So remember, a primary antibody is going to recognize your specific antigen. And more specifically, it's going to recognize an epitope on your antigen. And it's going to do that using the variable region of the antibody, which is made up of contributions from both the heavy and the light chains. So you have this variable region bind to your protein, and it'll still leave the constant region exposed. So kind of like the base of your Y is still going to be exposed, and that base of the Y is going to be constant. It's going to be constant for the animal that made the primary antibody. Therefore, once we kind of wash off the antibodies that aren't bound so we don't get more noise, we can add a secondary antibody that will recognize the constant region of our primary antibody. And this is then going to allow us to detect the presence of the primary antibody, which is telling us the presence of our antigen, because our secondary antibody is going to be conjugated or like covalently attached to something that we can allow us to see it, something like a fluorophore or something like an enzyme that we can link to a chemical reaction that will give off light. We'll then wash off the antibody that's still remaining, that's not bound, so we don't get even more background signal. And then we can actually do things like measure the fluorescence. So we excite it and measure the fluorescence in the machine. Or we can add a chemical that will then allow us to get light that we can measure, various things like this. I hate Western blots because there's so much washing, but it's really important to do these wash steps where basically you just kind of flow a buffer over the solution in order to kind of remove any non-specifically bound stuff, so that you're not detecting the presence of primary antibody unless the primary antibody is actually bound to the thing that you want. And you're not detecting the presence of the secondary antibody unless the secondary antibody is bound to the primary antibody, and that primary antibody is bound specifically to what you want. So there are a lot of wash steps to actually remove excess antibodies. And so typically you'll just have some sort of little box, and you'll have your membrane in that box, and you'll just keep changing the solution that you're flowing in and over the box. So it's a lot of washes, a lot of pain, and I don't like it, Western blots, but they are very useful. And so if you were to think about why, because they're allowing you to detect the presence of a specific protein and not the presence of all proteins. So thinking back to our SDS page, we can dye our SDS page gel with an all protein stain like a Kumasi. Here we can see that if we have a nice pure protein sample, that's pretty nice. You get this one nice band. If you have a lot of different bands, however, you have this kind of smear. You have a lot of different bands, and you can have bands that are kind of overlapping. So although this protein band right here, well, maybe that is the protein we're looking at here, maybe it's actually a different protein that just happens to be a similar size. And we can't even tell for sure that this is the protein we're interested in, because right now we're just seeing that we have this one pure protein. We don't know, is it our specific protein? We can get a good idea based on its size and based on how we isolated it, but we can't know for sure. a Western blot is going to allow us a way to tell what this protein is. Only, however, if we know what to look for. So an antibody for Waldo's hat cannot tell us if Wanda is present. So it might be that Wanda's present, but we don't know if Wanda's present because we're not testing if Wanda's present. And so if we think we know what this protein is, we can use an antibody for what we think it is and detect it. But if we use an antibody for something that it actually isn't, well, now we're not going to get any bands because the Western blot is only going to show us the presence of that specific thing. So this doesn't actually correspond to the specific gel, but with the Western blot, you're going to see very specific bands. Immunoblot is another term that you're going to see used often for a Western blot. And immunoblot is a little more generic. Sometimes on a gel, you might see like IB for immunoblot. That would be what it was talking about. So you might see WB for Western blot or IB for immunoblot. And you're not going to see all the bands. Hopefully you see very specific bands. But so the gel is going to tell you kind of what's in the sample. And so it's really helpful to know, like, is this a pure sample? Because if you were to see a Western blot and you were to only see these specific bands, you might be like, oh, that looks like it's really pure. But this Western blot tells you nothing about purity. The COMASI, the total protein, this is going to tell you about things like purity, but the Western blot is going to tell you more about identity. And it can only tell you the identity if you know the identity to ask for. It's like a game of Go Fish. If you don't ask for the right thing, you don't know if that thing is present. So you can't see what else is in the sample. You can only see if the specific thing you're looking for is in the sample. But it can be very, very sensitive. So for example, here what you're looking at is a total protein stain and a Western blot that actually do go together. And so the lanes, I don't know why they did this, but they kind of shifted the lanes over. So in this side on B, they actually labeled their ladder as 1. And then here they don't label their ladder as 1. And so if you think about shifting these numbers over, you would have lane 2. This is going to be 2, 3, 4, 5, blah, blah, blah. Bottom line is they take these different samples and they spike them. So they add in his tagged protein. And so this is the protein right here, this lane 10 or lane 9 over here. This is just the protein itself. And you can see that the band runs about here on your total protein stain. If you look at these lanes, you can see that there's a lot of kind of stuff in that actually also is that similar size in these samples. So only some of these samples actually have the his tagged protein in them. So only these, like this one and this one and this one and this one and this one have it. But if you say compare lanes 8 and 9, it's really hard to tell that this one has the extra protein in it. And so if you were to only see this total protein gel, that would not allow you to confidently say whether or not the protein was present. However, if you do a Western blot, in this case, they could do a Western blot using antibodies that recognize the his tag. Well, in that case, they're able to specifically detect which lanes have the protein present, as well as to detect kind of the abundance of it. So you can see that maybe some of these have a higher amount than others. Although without a loading control, you can't really know. And so we'll get into this in a second. But this is just to illustrate that you can detect the presence of even very small amounts of specific proteins, even if those specific proteins are similar in size to other proteins that are present in the sample. Another nice thing about this example is that here they were using an epitope tag. So in this case, they're using a his tag. Because we have a his tag on a protein, say for purifying it, we can also use an antibody that recognizes the his tag, save us money. Rather than having to recognize the protein itself, we can recognize the tag that's attached to the protein. And so that's what we'll see here is that they're going to use an anti-his antibody in order to detect the presence of their his tag protein. Often, however, rather than just detecting the presence of one thing, you want to detect the presence of a lot of different things. And typically, if you see like a cell biology paper, they're often going to be doing a lot of Western blotting. And most of the Western blotting that they do is going to be for proteins that are present, like natural proteins that are present, not modified proteins. So not epitope tag proteins, because one of the downsides of epitope tag proteins is that you have to actually go and genetically modify the protein. There are ways in which you can do it with CRISPR and things where you actually make the change in its endogenous location. So you add the tag to the sequence of the protein in the genome where it actually is. but that can be like this long, complicated process. And if you want to do things more simply, you can add a version into the cells. So just kind of add an extra version that is going to therefore have the modification, but then it's going to be expressed differently than the normal version. So there are a lot of complications that come into play if you want to introduce an epitope tag and not affect the normal cellular functioning. But epitope tags can be great in a lot of situations, especially if you're just kind of playing around in lysates in vitro, things like this. Inside of cells, things get a little more complicated, but there are methods that people will use. But it's also because there's some sort of common proteins that a lot of researchers are interested in, these kind of high button proteins, these things like oncogenes and oncoproteins, which are involved in cancer, various proteins that are involved in cell signaling. These are often going to have antibodies that are commercially available. A lot of researchers are going to use them to study these things. And so not only do you have antibodies that will recognize various proteins, such as your AKT, your MEK. So your MEK, these are like from your signaling cascades. You can see that in our signaling cascades, we have like phosphorylation of these kinases that then phosphorylate other kinases that then phosphorylate other kinases. And you can have antibodies that recognize both the normal and the phosphorylated forms of it. So there's lots of antibodies available for commonly studied proteins. And researchers will often want to study the presence of all these different proteins. And so you often see things that look all complicated like this. In this case, you have this one kind of membrane and it's showing you the whole membrane, but it's only showing you like this one band. You can imagine that, say they knew what they wanted, they knew the identity of one of these proteins up here. They could use an antibody to detect the presence of the protein up there. But things get complicated if you want to detect the presence of that other protein directly in the membrane at the same time. There are methods where you can like multiplex, where you can use antibodies at the same time. Even if you do that, however, your proteins are going to be like different places on the gel. And so often what scientists are going to do is they're actually going to cut strips out of their gel. They're going to know where the protein that they're interested is, and then they're going to cut their gel into strips, and then they're going to probe the strips to see whether those specific things are present, and then they'll show you the different strips. So you'll see something like this. Often these days, authors will actually have to show you their full gel in like the supplementary materials, but you'll see the strips just in the kind of general text. Another reason for doing it kind of like separately is different antibodies will have different sensitivities. And depending on how much of the different proteins are present, you might be like overexposing one of your samples and underexposing another if you were to try to measure them at the same time. The strength of the band in the Western blot can tell you about the relative amounts of the protein present. We'll get into some caveats about how different antibodies have different sensitivities. But if you were to compare the same amount of sample loaded in different lanes, and you see that one of the lanes has a fatter band for one of the proteins, that would tell you that there was more of that protein in the sample. In order for this to be true, what needs to be true is that you loaded the same amount of total protein in both of the lanes. If I see a fatter band, is that because I have more of the protein present, or is it because I loaded more of the total protein? This gets to the idea of loading controls. Housekeeping genes or housekeeping proteins are going to be proteins that are expressed at kind of similar levels in most cells. Some common ones you might see are GAPDH, so glyceraldehyde 3-phosphate dehydrogenase, a core player of glycolysis, as well as actin and tubulin, which are key components of the cytoskeleton, holding the cells together. And so these proteins are going to be expressed at similar levels, kind of ubiquitously, so all the time, everywhere. And therefore, you can use it as a normalization factor. What you can do is if you detect the protein, at the same time you're detecting your samples, you can then divide the signal from your protein by the signal for that GAPDH protein or the actin protein or whatever else you use in order to kind of have a normalization factor, to have a point of comparison. This is really important because the signal that you're going to see is going to correspond to the amount of protein. And it's not going, and if you load more protein into your gel, you're going to see more signal, even if there's not more protein per cell or per volume or per concentration. So for example, often what you're going to see is you're going to see the results depicted in bar graphs shown below the bands or above the bands. So you'll just see a representative bands. But often what scientists are going to be doing is they're going to be running multiple trials of this, and they're going to be measuring this amount of signal. They can do this using densitometry, where they just use a computer to calculate the kind of amount of signal in the various region of the gel. So you stick a little box around the band and you say, how dark is it in the box? And then you can compare the darkness in the different boxes. And you're often comparing the darkness in your box compared to the darkness in the loading control in order to normalize. So your loading control, you have your housekeeping gene, your thing that's ubiquitously expressed. You then detect the presence of it in all the samples that you run so that you can divide the signal from your protein by the signal for that control in order to figure out if your protein is really being expressed more, if your protein is really present more in those cells, or if it's just that you loaded more in the gel. And so for this example, we can see that they have loaded kind of more of their sample, they have, or at least they have more of their tubulin in some of these wells than in others. And so they need to take this into account when they measure this. So for example, if we were to look at this sample right here, compared to this sample right here, in this sample, you can see that we loaded a lot more. So if we look down, this is going to be corresponding to our sham MIR, myocardial ischemia reperfusion. So they're looking at treatments for heart problems. So they treated with adiponectin, and they wanted to see whether that influenced the biomarkers for various bad things. And so if they don't add the adiponectin, they see this increase in this gene, GP91-FOX, and this INOS. And so they're seeing if they add adiponectin, does that go down? And they also have these kind of mutant cells that they're testing as well. And so we're not going into the details here about their experimental questions, but rather how do we interpret their data? We can see that here they have loaded similar amounts of the sample in all of these wells, and so we can kind of more directly compare these bands. However, in these mutant cells, we see that they seem to have loaded more of some of their samples than other samples. And so we need to, in order to fairly compare all these samples, divide by the amount of their loading control, divide by the amount of the beta tubulin. So we would measure how dark these different squares were, and then we would divide the density of those. So for example, if we were to look at this here, where we have sham treatment of the wild type versus sham treatment of these AMPK-DM cells, we can see that it looks like we have a lot less of this GP91-FOX in our sham than in our wild type, but we can also see that we loaded a lot less of the sample in those cells as well. And so if you were to go and look at the actual quantification, we can see that they're actually very similar. And so we needed to actually, however, be able to normalize in order to tell that. So in order to actually be able to compare between samples, you need some sort of normalization. This is going to then allow you to know that, yes, there were similar amounts in these cells, but this cell compared to the wild type, there's actually going to be a lot less, because we're able to see that in comparison to the thing that we know is expressed to similar levels, we're going to have a lot less present in our sample. Now, this is assuming that the housekeeping protein that you choose is actually going to be constant. And so there are technicalities where sometimes those proteins aren't as kind of like constant as we would think. There are methods to kind of quantify based on multiple housekeeping proteins, quantify based on total protein, or at least test multiple proteins to make sure that they are consistent through your cells. One way or another, you need some way in order to normalize your samples, in order to know that if you see more of a sample, it's because you have more of that protein present, not because you have just loaded more of it on your gel. Even with the normalization to the loading control, we still have the problem that different antibodies will have different sensitivities. And so you can get different signals for the same amount of protein if they were different proteins and the antibodies had different sensitivities, and maybe you exposed one longer than another, all these things that are going to prevent you from comparing different proteins to one another, but you can compare the same protein in different samples. And in order to compare between samples, however, you need to have the loading control and be able to normalize to that, or to total protein, one of those other strategies. Let me illustrate in a little more detail. Western blots are kind of semi-quantitative, And you can do things to make them more quantitative. Because antibodies will have different sensitivities, you could have like 10 mg per mil of one protein A and 10 mg per mil of protein B. And you add antibodies against protein A and antibodies against protein B, and maybe you get twice as much signal for protein B, but that doesn't mean that twice as much protein B was actually present. If you were to run protein B, but you only had 5 mg per mil per sample, well, then you would get the same amount of signal as if you had 10 mg per mil of protein A. And so you can't really directly compare between different proteins in terms of quantitatively, but you can compare the same protein in different cell samples. And if you were to run like standards, so you know how much protein corresponds to how much signal, then you would be able to quantify it a little more. But just having a normalization factor for your loading, like your GAP-DH, is not going to address the problem of these antibodies having different sensitivities. But it is going to allow you to compare the same protein in different samples, but not different proteins within the same sample. Unless you're just kind of doing a comparison that's the same in each of these different wells. So if you want to say like, what's the ratio of this to this in terms of signal, you can do that, but not in terms of actual quantitative numbers. So Western blot, you have to run a gel, transfer to a membrane, and probe the membrane to see what's there. There's a similar method that doesn't require all of that gel and blotting. It's called an ELISA, or an enzyme-linked immunosorbent assay. And there are different varieties of it that can be used to test either for various antigens or for various antibodies. And so I'm not going to go into the details, but in what's called a direct ELISA, you would be taking your sample, so that contains the antigen or potentially contains the antigen, so some protein you want to look for. And then you're going to actually bind those to the bottom of the well. And then you're going to put an antibody that binds to that antigen and is conjugated to an enzyme that when you add a chemical, it will produce a signal. But in between kind of adding the antibody and adding the chemical to make the signal, you're actually going to wash the wells, and that way only the antibody that's actually bound to the antigen will be present, and the antibody will only be bound to the antigen if the antigen was present, and so that way you can kind of detect the presence of that protein. There are other methods in which you kind of have an antibody bound to the plate, and then you have your antigen bind to that antibody, and then you have another antibody that then binds to the complex of the primary antibody bound to the antigen. And so this is an example of a sandwich ELISA. So it's just another method. This case too, you're detecting the antigen. There are also other methods, however, where you might use in medicine to actually test patient samples, so like blood samples, or at least the cell-less part of the blood samples, so like plasma or serum. In this case, what you can do is you can actually have, say, a viral protein bound to the wells of the dish, and then you flow a patient sample over it, and you're trying to see whether the patient has antibodies that will bind to that antigen, that like viral antigen or whatever disease. And then you're going to add a secondary antibody typically, which will bind to that primary antibody and be labeled. And this is going to therefore allow you to see whether the patient had antibodies that recognize the protein, which would indicate that the patient has been exposed to that virus or whatever, and that they have mounted an immune response against it. So it could be something that they had in the past or something that they had in the recent past, but that would be a way that you could measure that. And you can even quantify an ELISA if you have like a standard curve of what the antigen is or what the antibody is, whatever it is that you're testing for, see how much signal corresponds to how much of that protein present. So it's simpler than a Western blot. You don't have to go through the gel and everything. It's easier to run a lot of samples. You can quantify it more easily. However, you're missing the size information. And that size information can be really important because you could have your antibody be recognizing something non-specifically, or you could have the antibody be recognizing a lot of things. And then any one of those things could set off the signal in ELISA, and you wouldn't know whether or not it was actually the thing you were looking for. At least with the Western blot, you're able to see the size, so you know that if you compare it to a ladder, yeah, that's the size I would expect this to be. Whereas with an ELISA, you just see, oh, there's a signal. And yes, you can say exactly how much signal there is, but that's not going to tell you if that is actually the protein you think it is. And so typically you want to have a lot of validation for your ELISA that you are using very specific antibodies, and the antibodies you use need to be very, very specific. With a Western and an ELISA, that'll tell you about the presence of a protein. But if you want to know about interactions, there's a technique called a Co-IP, which is a form of a pull down. And in these assays, you're basically able to pull out or remove a protein from a mixture and then look and see what it's bound to, such as using a Western blot. And so more on that in the next video. So to review, antibodies are little proteins that recognize specific parts of other molecules, such as specific parts of proteins. We can take advantage of the specific nature of their binding in order to use them to detect the presence of those proteins that they recognize or other things that they recognize. We can do this in samples that we run through a gel and transfer to a membrane. This is the case of our Western blotting. We can do things in cells and staining cells, which would be an example of immunocytochemistry or immunohistology. And we can do things like do this in wells of a plate. That would be the case of an ELISA assay. If we want to get fancy, we can talk about using co-immunoprecipitation, where we use antibodies to pull out or kind of isolate from a sample a specific protein, which we would call a bait, and then see what the protein was bound to, the prey, using a Western blot. A key thing about Western blotting is that it can tell you if specific proteins are present, but only if you know to test for them. If you don't know to test for them, you won't see that they're present. And so there might be a lot of proteins present, but you're not going to see them unless you specifically test for them. Like a game of Go Fish, you're not going to be able to know if the other player has a card unless you ask specifically for that card. The way that we ask specifically for different proteins is by using different antibodies. Because there are so many proteins that people want to test for, it can get very expensive. And one of the ways in which you can kind of save money is by having epitope tags, where you add a little sequence onto the end of your protein that's going to serve as an epitope, or the thing that an antibody recognizes. We can have things like HA tags or MIC tags, V5, His tags, various things that you can then buy cheaper antibodies that recognize those tags. And as long as that tag is on your protein of interest, you'll be able to indirectly detect the presence of your protein of interest. But that's going to involve some sort of genetic engineering to add that tag on. You might be adding extra versions of the protein and kind of just ignoring the version of the protein that's normally made. Things get complicated if then you're wanting to look at expression, but if you want to look at interaction, that could be a good strategy. But if you're overexpressing something, you might see interactions that aren't very natural. All sorts of complicating things that you can get into if you really think about the practicality of putting this into use. But the basic theory is, at least hopefully, understandable and very useful.